All right, great, thank you. Good afternoon or good morning, wherever you happen to be. Welcome to the Benthic HABs Workgroup webinar. I'm Margaret, one of the facilitators, along with Jade and Christine. And we're going to get started, and I'd like to remind everyone to put their phones on mute so we can clearly hear our presenters. If you have questions, you can wait till the end of the presentation where we're going to have short discussions. And also you can use the chat segment. We'll try and check that for questions. Next slide. So the presentation today is going to be posted on the EPA's website, along with the presentations from our past webinars that you can look at. Also, how to contact the 3 facilitators of this work group if you need to be added to the distribution list or you have questions for us. And then any information on future webinars as it becomes available will be posted there. Next slide. So on the agenda for today's webinar, Jade Young, one of our group facilitators, will review the results of the 2019 Benthic HAB Workgroup member survey that we had a great participation in. And we're pleased to have Keith Bouma-Gregson as our guest speaker, who will present on Cryptic cyanotoxin producers in benthic mats. Keith is an environmental scientist at the California State Water Resources Control Board and co-lead of their freshwater harmful algal bloom program. His primary interests are in the ecology of benthic cyanobacteria and their effects on water quality and public health. He received his PhD from UC Berkeley in 2017 researching benthic cyanobacterial mats in the Eel River in Northern California. Finally on the agenda, Christine Joab, another group facilitator, will guide an open discussion for the group on the latest and greatest in the world of Benthic HABs and plans for the work group in the future. So without further preamble, Jade will take us on to our group survey results.
Yes, happy to do that. I'm Jade and a co-facilitator, and my regular job is at the Army Corps of Engineers in the Louisville district. That's Louisville, Kentucky. And today I'm going to be talking about the Benthic HABs discussion group and the survey of the membership that we conducted. But if you ever need anything from the Corps or have any questions, my contact information is there. Don't ever hesitate to contact me to ask. The mission statement of our group, mission of this international collaborative, is to accelerate the mutual understanding of benthic HABs in rivers and lake systems by sharing data and monitoring protocols, experiences, and lessons learned. And so far, I think that we have definitely done that in this group. We have 3 facilitators, and we've introduced ourselves already, but you will have our contact information here and job titles, etc. Yes, as we mentioned earlier, this presentation will be posted and also did go out to the membership if you are on our membership distribution list. Also, I meant to mention that if anyone wants to volunteer to be a co-facilitator and work with the 3 of us, we would be happy to welcome you to that. It's a good opportunity to diversify your resume or just meet some new people. Prior to today's meeting, we have had several meetings and a diverse group of people that have presented to us. It's been a good variety, and here's just a summary of some that I've picked out here to point out to you all. And the point of this slide is to just give you all an idea of what's going on and what we've talked about so far. And I can tell you that I have definitely learned a lot from each presentation and each person that shares something with the group. We did conduct what we called a re-survey of the membership, and it was just a second survey of the people we had on our list and an attempt to grow our membership a little bit. And and I'm going to review that right now, and I think it's best if we keep this discussion informal because there are some questions in the survey that we would like to discuss to get some more information to provide direction. So we did receive 40 responses to the survey and had approximately 17 to 22 new members as a result of that, and our regular membership is approximately 100 people. So that's pretty good, and we did have several new agencies represented on that, and I just want to say welcome to our new members. And for all of you that did respond to the survey, we really do appreciate that. And the point of the survey is just to stay relevant and make sure that this is a member-driven group. So the first question in the survey, and we want to discuss this just a little bit, is just a question of what role is of most interest to you. And most people responded that they wish to be a passive participant, and that's great. We definitely want a lot of. If anyone wants to step up and add to your resume, we would appreciate assistance in active participants that could share information or in co-facilitators. Again, I'm not going to be able to message you guys on here and present at the same time, but I will check that later on. So part of this question was the question about networking. A lot of people, 7 people, responded that they wish to create subgroups specific to the areas of interest. So I would like to find out from you all, what are the areas of interest that you had in mind? Anybody want to volunteer to speak up on that? Anybody there?
Hi, this is Elizabeth Hilborn. Can you hear me?
Yes.
I'm really interested in health effects associated with benthic algae. So I'd be interested in a work group or subgroup in that area. Animals,
Okay.
Animals, people.
You heard my name.
Okay, I've got that written down.
Hi, I'm Bree Olson with Arkansas DEQ. I'm really interested in knowing how to sample for this kind of thing.
Do you mean, do you have anything a little more specific, like sample collection or?
Yeah, more like collection. Yeah, because, we'll do just regular cyanobacterial blooms that you'll get on ponds or lakes, and, I think we know kind of how to sample those, but it's not as intuitive when you've got blooms on the bottom of rocks or, substrates like that.
I would second that interest.
Another interesting topic would be the food web impacts of benthic cyanobacteria.
This is Keith here in California. One of the things we've been talking about is messaging for the public and how to post and put signs up when the cyanobacteria is benthic rather than a planktonic bloom. We have a lot of the messaging sort of have a better handle over it when it's a bloom, or a planktonic bloom, but not a mat. And so I would enjoy maybe working with folks in other states who might be thinking about that and talking about messaging.
This is Bob Clement. One of the interesting things, and I don't know if it's related to the whole benthic work group or not, but there's some recent discussion about the algae cysts that are off the coast of Alaska, and they're down at the bottom of the ocean, and I don't know if there's a lot of research to what constitutes what conditions will make those cysts reemerge and really impact the food chain. Just throw that out there for.
This is Kurt Carpenter in Oregon. Another area that I think could deserve a focus group would be remote sensing for benthic HABs. I mean, of course, the cyan and other programs are looking at lakes and reservoirs of certain sizes, but I don't think it'll be too long before hyperspectral monitoring will be able to identify benthic growth of algae and or cyanobacteria in larger sized rivers. That'd be worth looking at.
Okay. Got that down.
Yeah, this is Bob Clement again. When you look at something like the Platte River in Denver, you don't see the typical algae mats. What you do see is a lot of benthic algae, and so nobody has aspired to do any type of sampling because it doesn't fall into the traditional. There's algae blooms there, so they don't sample, and so they don't. Even though there may be a lot of benthic algae, they're not actually sampling to see if there's a cumulative impact from the toxins.
From that standpoint, I know the Platte River was one of the major river systems that was studied for about 20 or 30 years as part of the USGS National Water Quality Assessment Program, NAQUA, many of you are probably familiar with. But doing a query of those data nationally is a big job, but I could see where a compilation of looking at for Oscillatoria, Formidium, Nostoc, many of these benthic forms would be a really good first start to get a national picture of this if something like that's not already been done.
Yeah, it would be also. This is Bob Clement again. That would also be interesting to see, how the samples were taken, what their frequency was, maybe even what time of the day the samples were taken to kind of, put that sampling in perspective as to what it might have captured and what it might have missed as far as sampling frequency and timing and stuff.
Yeah, I mean, unfortunately, most of those. Because I collected a lot of that data in Oregon. It's just a one time per year. So, but at least that would give us maybe a sense of the major genera and the types of rivers they might grow in, and at least kind of a presence indicator.
That would be great.
Yeah.
This is Todd Edgerton from Virginia. I think we'd be interested in really a better understanding of how to relate the health impacts of these benthic mats compared to water column cyanobacteria as it would relate to ambient or recreational water quality criteria. How we should be looking at these.
All right, I've got that down. Sounds like we have a lot of groups and a lot of things to work on. That's great. Really appreciate everyone speaking up and saying something about that. Is there anything else before I go on?
Yeah, I have one thing. Are there any plans to convene all these folks together for a national type of a meeting? I mean, I think if that hasn't happened yet, that would be a good thing to do.
There's nothing that I'm aware, and that we can put that on the list of topics to discuss. All right. Well, the next question was, do you have an area of interest or expertise? And as I was reviewing the results, I realized that this could have been misinterpreted. But the real purpose of this was just kind of get an idea of kind of the demographics of our group and what everyone was working on. So either way, this is good information and good feedback for us. Most people are interested in monitoring and data collection. Second to that was research. And then after that is regulatory thresholds, for cyanobacteria and cyanotoxins. And of course, we're dealing with something that's sort of newer, like this issue. Those are definitely going to be big players. So it's great that we have a membership that is interested and involved in those areas. We did allow a other category where you could type something in, and this is these are the other comments we received. Water treatment and impacts to treatment, any information that would assist reservoir projects, water quality modeling, biomass and fluorimetry of phycocyanin, policy mitigation, drinking water treatment for cyanotoxins, and environmental drivers. Anybody want to comment on that? Does that adequately capture everyone's interest on the phone, or do you feel that you would like to add to that?
This is Bob Clement. One of the important things that we don't know about benthic algae and algae in general is we haven't discovered the mechanisms, what environmental conditions actually trigger the metabolic machinery to produce toxins. And until we know that, until we get some type of idea what that is, then both the algae that's floating on the surface and the benthic algae can be potentially at risk for producing a high level of toxins. We may not know, we may not have experience in the past where benthic algae had contributed to a large production of toxins, but until we know what the mechanisms are, what stressors on those benthic algae may cause them to, have an outbreak of a lot of toxins. And, so I think that's an interesting subject that really we know very little about.
Dave, quick comment on that. And you're right on there, Bob. I mean, I think that this occurred again. This is the big question that hopefully a lot of people are working on. I'm aware of a couple researchers that are doing this, but that's another, I think, work group, not just from the benthic side, but also from the planktonic side. Just a couple things to share if you haven't seen this, but I was at a Region 10 EPA meeting recently on HABs in Seattle, where we were reminded, because this was shown in some studies from the 2015 meeting as well. So this has been around for a little while, but that cyanobacteria grown on urea seems, for some test species, seem to produce more toxin than grown on other forms of nitrogen, for example. And so that's something that I think we should test with not only more planktonic species and experiments, but also looking at the benthic, and then the other part of the interesting part of the story that came out of Susie Wood's work that I was seeing, looking over her presentation from her cyano HAB work from the benthics, was that reaches with low nutrient concentrations tended to produce formidium that was producing a lot of toxin. And so that just raises all kinds of implications for me and for these Oregon rivers, where we have reaches that are very nutrient enriched with lots of benthic algae, but then alternating with reaches downstream where the nutrients have disappeared. And that potentially opens up a niche for these benthic cyanobacteria to get in there with all their different energy strategies that might not necessarily require a bunch of nitrogen from the water column per se. So getting to those environmental drivers of toxin production is critical, and that would be, I think, another super important working group to establish and get the different biochemists together so that they're learning from each other. Thank you.
So another question was, what are your information sharing preferences? Most people marked webinar, and that's what we're doing today, and that's what we've done most of the time. And then next after that was the subgroup meetings. We've got a lot of ideas for that already, so that's great. What about these web-based platforms? What are they? Does someone want to talk about that any? All right, well, that's okay. If you think of something and want to send it along, just go ahead and do that. And then we had meeting frequency. Most people said quarterly. And then someone else marked just as needed. So one thing that we will be discussing and pleading for are presenters. We do need people to present. The past couple of times we've had a meeting, we have had quite a bit of difficulty finding someone to present. And you know, if there isn't a lot going on or people don't want to share that through the webinar, that's fine, but we may have to reduce the frequency of our meeting. So,
Excuse me, this is Elizabeth. Do you need a presenter for every meeting? Are there other discussion topics versus a presentation?
now there are, because we have these subgroups, yes. You know, I guess that's just not something we thought about, was to just have a meeting to discuss different topics. So that's a great suggestion, to branch out to more just like a free-form discussion.
Yeah, this is Bob Clement. I definitely second that. I don't feel that there's a need to have a presentation at each one of these quarterly meetings. I mean, if there's a list of topics, I think that's sufficient.
Okay, great. So what would that look like? Would it just, will we just send out a topic and then we just call in and just everybody share their information, or would someone be the lead to formulate questions ahead of time?
I think it would just be good to just put out to the work group a couple of weeks beforehand. What agenda items would you like to discuss during this next meeting?
This is Elizabeth again. May I suggest there are multiple models we could follow. One would be a journal club type of approach where we send out a couple of papers, maybe with some unifying theme, or a single paper to discuss and talk about maybe how the results could be applied in our area. Because we're not just from California. When we were talking about a estimation of Benthic HABs across the country, I was thinking, how would that look? In North Carolina, we have so much sediment pollution. I hardly see rocks at the bottom of our water bodies. So, you know, maybe to talk about the different things we're seeing and dealing with a journal club type approach, or another type approach might be, I've seen the model where just people are polled across the membership of that meeting about any topics they want to bring up or discuss or bounce off other people. So I think there are multiple models to choose from.
Thanks. I had never heard of a journal club or something like that, so that's great. Thank you for that.
This is Keith. Adding to that, if there is the interest in these subgroups, which the subgroups didn't exist in the past, it could also be that subgroups are tasked with reporting out on their progress or given a update on their objectives, and they could maybe be the leaders of a meeting of the particular topic they've been working on.
Okay. Well, here's maybe the biggest question, and the one thing that can affect our forward movement here. This question just asks, what are your top 5 categories of interest that you would like to see in this work group? And the top response was water quality drivers. After that was technology and using technology to identify blooms. And then we had monitoring and data collection. And then tied for 4th and 5th were toxin species relationship and treatment. So does anyone want to get more specific on this? Maybe you selected one of these options, and we haven't, brought it up yet for one of the subgroup ideas.
Looking specifically at one of the cyanotoxins, namely Anatoxin, might be worthwhile. Some systems might not have issues with Anatoxin, but many others may, including out here in Oregon. And I know the big emphasis has been on microcystins and cylindrospermopsin, especially with the drinking water guidelines being issued by EPA on those. But more work is needed for Anatoxin, and I think having a focus group on maybe all the neurotoxins, so Anatoxin, saxitoxin, which we also have here, freshwater and marine, would be something to consider.
This is Angela Shambaugh. One of the reasons I joined this is because we've had very few experiences with Benthic HABs on a large scale. We see them reported from small ponds and things like that, not infrequently, but just having an opportunity for folks to share their experiences and photographs of what you've experienced and how it changed over time in a real informal setting would be really beneficial for me. I'm kind of a visual learner in that respect when it comes to taxonomy and keying into cyanobacteria, so just an opportunity to share photos and experiences in a more informal way would be very helpful.
Along those lines, the taxonomy, I know you have it listed here as an interest, but I think that given the pretty quickly evolving state of cyanobacteria taxonomy in general, that special attention to benthic taxonomy of cyanobacteria is worthwhile.
I would say there's a great interest in that. This is Leah from ODU.
So can you guys elaborate a little bit more on the taxonomy? Are you more inclined about the different tools and technology to differentiate in the taxonomy, or are you interested in getting like a information on the different types of benthic cyanobacteria, the taxonomy associated with identification in that?
Yeah, well, I think it's kind of all of the above. Most of us are used to looking under a microscope and picking out, certain morphological features to arrive at our best guess at what it might be. Having an integrated approach with the genetics, of course, would be, I think, an important part of it. So, looking at it from all different angles, so that, ultimately it would be really nice to be able to identify by sight, in the field macroscopically, and just quick response to the comment from North Carolina with the sediments. Those sediments, a lot of times you'll see a lot of benthic, cyanobacteria, oscillatoria, formidium, and such growing on the margin. So you might not need to get all the way out to the rocks to see them, but being able to make the connection between what you see under the microscope and then, ultimately one of the key things is it a toxin producer? And so, that's going to, I think, take quite a bit of work. But it'd be nice to be able to move that ball down the path some, with the taxonomy from, the genetics and the morphology.
One of the things we've seen here in Vermont is that even a species or a genus, I should say, because I hesitate to identify things as species, even one that you think you recognize macroscopically can change. We've got one particular genus that has manifested so far in 3 different ways. So it would be, again, trying to understand the taxonomy and again, connect it back to whether it's a toxin producer or not. But building those search images and being able to communicate them is really important.
What genus is that?
Anabaena.
Okay, thanks.
Yep.
We have a lot of that in Oregon too.
Yeah, seems to be a recent, I don't know if I can say it's a recent introduction, but we did not see it in our larger Lake Champlain prior to 2012. And now we see it in the plankton, now we see it in the benthic benthic areas, forming mats that float at the surface, floating scums that look like planktonic forms initially, and so it's got a variety of presentations, which has been of interest, and I have no idea whether that's normal or maybe we're not identifying things correctly.
Interesting, thank you.
What about this technology and using technology to identify blooms? Is that a reference to remote sensing?
Yeah, I think that's true. I think the hyperspectral capabilities on microscopic cameras is something else that will, maybe eventually be available.
I also believe people are doing some work with kind of like an algae torch sort of thing to look at benthic as well as planktonic.
Did you say algae torch?
Yes, I think that's a brand name.
Okay.
Yeah, the bentho torch, I think they call that.
Okay, I've heard something about it. I just don't know the details of it.
This is Bob Clement. So, can benthic algae give us some type of correlation to the health of a water body? I mean, I typically see the benthic algae in streams. And so, you look at the plat and then you compare that to, the water coming out of maroon wilderness area near Aspen, and you can take a picture down at the rocks in the stream. And if you don't have any ripples, you can't tell there's any water above it. And you look at Gross Morn National Park in Newfoundland, and the lake there has got so little nutrients in the water that there is no benthic algae in there. So, is there, can we consider, the development of the levels of benthic algae in the stream and what we visually see as some level of determination of the health of the stream, the impact, from agriculture, nutrients, etc. Because we see such diversity as far as, from nothing to different levels of benthic algae. So can it be helpful and helping to discuss the health of the ecosystem and the stream, and maybe even the, when we get to a lake, the algae blooms that might occur?
All right. Well, guys, it looks like I am out of time. But we'll be following up on these things with you guys and sending out some calls for who wants to be in what group, and maybe even continuing this more specifically later on. And just future directions, I think I already discussed that. And don't hesitate to contact us. And thanks, everyone. And Keith, I will...
You guys starting to see my presentation?
Yep.
Yep.
Okay, great. Okay, and now it's in full screen view for everybody?
Yes.
Great. Wonderful. And the audio is okay?
Yes.
All right. Home run. Great. Well, thank you, everyone, for attending this, and also thanks to Christine and Jade and Margaret for giving me the opportunity to present here. So my name is Keith Bouma-Gregson, and I, as mentioned before, I co-lead the Freshwater Harmful Algal Bloom Program for the state of California, and I'm going to be sharing about some of the cyanotoxin, our work to understand who's producing cyanotoxins in these benthic mats. And some of this stuff was work I did as part of my PhD dissertation, and then the bulk of it will also be work that was done a couple years ago with scientists at the Cawthron Institute from New Zealand, and Laura Kelly was one of those scientists, and she took the lead on synthesizing the data that we collected, and so a lot of the figures and things that I'll show in a little bit were primarily generated by her, so I want to make sure I give her ample credit for some of the work you're going to see here. Great, so where we're going to take a journey to is to Northern California, primarily the Eel River, which is a 10,000 square kilometer watershed that's up in the coast ranges of California. So north of San Francisco, it takes about 3 hours by car to get up into the watershed from San Francisco. And it's a pretty rural watershed. The traditional land use was forestry and ranching, and then more recently cannabis production, which has created interesting social and environmental nexus for marijuana affecting the landscape. Like I said, low population density and pretty much just 2 dams in the watershed and pretty high up in the watershed. So unique. This makes the eel pretty unique for a California watershed to have such little hydrologic modification. So most of it is flowing as a natural flow regime. In the southern portion of the watershed, which is the upstream portion, is the Angelo Reserve, which is a field station that UC Berkeley operates, and that's where I based a lot of my sampling out of when I was a graduate student there, as well as there's been a lot of other work on algal ecology done there over the years. And another important thing to note about the Eel River and California in general is the Mediterranean climate. And so you can see there in 2 of these images, I show a photograph of the eel in March where the flow was about 1,500 cubic meters per second. And so our winters, we get storms coming in off the Pacific, which raise the river levels, and the Eel is a relatively flashy river. And then in late spring and early summer, the storms stop, and we go through usually a 5 to 6 month period without any rain, and then the river levels drop drastically. So by August, the river's flowing at about a cubic meter per second. And it's during these summer months that we get the warm, slow-flowing, clear water, and so we have a lot of benthic algal growth. And there have been a number of dogs that had died in the Eel River in the 2000s. And then also some other watersheds. It's about 15 dogs in the Northern California coast rivers dying due to benthic cyanobacteria. And so as part of my dissertation, I was interested in understanding some of the patterns and the organisms that were out there. It hadn't really been investigated that much in this region. And so we set up a number of monitoring sites and collected mat samples and measuring cyanotoxins over that time. And so I'll share some of that information here. And these photos kind of hopefully give you a sense of what the river looks like in the summertime condition. And so one of the main results was a pretty and very consistent detection and measurement of anatoxin across the whole watershed. And so when I started this research, there had been a number of dogs that had died, but it wasn't really known if which toxin was necessarily producing it. There was some reports of anatoxin, but we didn't have a sense if this was just like a really isolated incident or if it just was occurring in random years. But pretty much over the 3 years I collected data, we were picking up anatoxin across the watershed at multiple sites. And we were also measuring for microcystin, and very consistently the microcystin levels were much lower than the anatoxin levels. And so we concluded that anatoxin was probably the primary cyanotoxin of concern in the watershed, and that these data also supported, like I said, some of the anecdotal information that was collected around some of the dog deaths. And so in each of these maps, each of the black dots was where we were collecting. We have a monitoring site set up, and then the width of the dot is the amount of anatoxin we got in a given month. And so building off of that result, I was going around the watershed trying to understand where these cyanobacteria were, if you could see them. And 2 of the common mats that we saw, like genera that were forming mats, were species of Anabaena on the left here. And what we found is that those were pretty closely associated with green filamentous algae. So in those middle panels, C and D on the left, you can see that the Anabaena is growing epiphytically on top of Cladophora filaments, which is a non-toxic green alga. And this is pretty much how we see Anabaena growing. And it will make these distinct spires, which occur as the oxygen from photosynthesis gets trapped in the mucus and creates these bubbles, which then cause the whole assemblage to rotate vertically and pull towards the surface. And in panels E and F there, you're you're pretty much seeing that can be pretty much the dominant algal assemblage will be these Anabaena spires growing on filamentous cladophora. And these tend to grow in slow-flowing parts of the river, so it's usually channel margins, backwater pools, or later in the summer to mid to late summer when the flows in the river have dropped. Another taxon we see is Microcoleus, which also known as Firmidium, and I'll talk about taxonomy in a couple more slides and why I'm calling it Microcoleus and not Firmidium. But that is, you find if you've read any of Susie Wood's work or the work that's come out of New Zealand, this is pretty much the same genus that they have issues with there. And similar to the findings and patterns in New Zealand, this grows epilithically, so it grows directly on the rocks, and it tends to grow in faster flowing waters. So we usually see it in riffles and runs, which is in contrast to the Anabaena, which is in the slower flowing water. So these were pretty much the two species that we saw consistently creating large macroscopic proliferations of across the river. And so then we were also going out and collecting mat samples from these two mat types. And on the left, you can see a bar chart of cyanotoxin detection. So then this is in each of those samples. It's just, did we detect either anatoxin or microcystin? And in both the Anabaena and Microcoleus samples, in the majority of samples, we were detecting one or both of those toxins. So it was rare not to have a mat that had some toxin in it. And then on the right, the bar charts show the actual concentrations, and those are on a log scale, also to note. And regarding the concentrations, the average for Anatoxin, the average concentrations between the Anabaena-dominated mat and a Microcoleus-dominated mat were pretty similar, but we did see some of the highest maximum concentrations in the Anabaena-dominated mats. And then similar to those other spatial results on the maps, we found lower microcystin concentrations in our mat samples than anatoxin concentrations. And I'm realizing I forgot to mention in the slide before where I was showing the maps, that was all dissolved cyanotoxins in the watershed. So those were collected by looking at the dissolved toxins in the water column, and the data I'm showing you here is actually what was intracellular, so what was actually within the mats themselves. So it was nice that the patterns between the dissolved data and this mat data were lining up, and that we were seeing a lot more anatoxins, and that also supported the other evidence from dog deaths, which associated a lot of the dog deaths with anatoxin as well. And so then some other work that was done, that I collected a bunch of these Microcoleus mats and did some genomic sequencing of them. So we sequenced a bunch of mats and then tried to reassemble their genomes together. And what I found then was, when I compared each of these draft genomes, was that there was actually potentially 4 different species of the Microcoleus in the watershed. And so this figure on the right, each one of the little squares is a Microcoleus genome from a single sample, and they're each compared with one another. So it's a pairwise comparison, and then they're grouped together by how similar their DNA sequences are. And when you group them together, you see these 4 clusters come up, and so each one of those clusters is basically a species of genomes that are very similar to one another and would count as a species. What was also interesting from this is that all the reference genomes that are out there had very low similarity to these genomes that we found in the river. So there's some potential that these might be a novel genome, and we don't have a good reference genome for them that's sitting in a culture collection or somewhere else. One of the reasons, I wasn't going to go into this, but with some of the talk just a moment ago about taxonomy, as I was putting together this paper and trying to figure out what these genomes were, there was a paper in 2013 by a Czechoslovakian taxonomist, Otakar Strunecki, where he moved a bunch of Firmidium autumnale species into the genus Microcoleus. And when I was looking at some of the marker genes on from these genomes, they basically fit within the group of Firmidium species that Strunecki was moving to Microcoleus. And so I chose to go with that revised taxonomic distinction from that 2013 paper. So prior to this genetic work, I had been calling these species Formidium, but then post this work, I've been calling it Microcoleus. So would definitely enjoy furthering that discussion. If other people have molecular data on the Formidium or Microcoleus, it'd be great to talk more about that and see how this works, because it's as folks mentioned, taxonomy changes quickly, and then we can lead to a lot of inconsistencies. So that's a little digression on taxonomy, and then within that, of those species, we were able to look for the Anatoxin biosynthesis gene cluster, and only what we're calling species 2 had that gene cluster in its genome. So even though we potentially identified 4 different species, it seems like only one of them might be a toxin producer, and the others don't have the genes for producing the toxin. And then down there on the left of the slide, or the bottom of the slide, shows that gene cluster. And so that information there was all gathered from environmental samples and draft genomes. Working with a taxonomist in Southern California named Rosalina Sancheva, myself and some others sent her some of our samples, and she tried to get some isolate cultures developed. And this is also work that was done by Rich Fadness, who is one of the regional water quality staff, and he's the main harmful algal bloom lead for the northern coast region of California. And so once those cultures were established, we did tests for Anatoxin A, microcystin, nodularin, and cylindrospermopsin, and basically found that none of those cultures were producing microcystin, saxitoxin, or cylindrospermopsin. However, several of them were producing Anatoxin A, and so that's this table at the bottom. You can see that we have 7 cultures that from 3 different rivers where Anatoxin A was detected in them, but then we also additionally had an additional 7 Microcoleus cultures with no Anatoxin production or detections. And so that once again supports that genomic work, which was showing that we have toxin and non-toxin producing species there. What was also reassuring was that one of the toxin producing species from these cultures was basically the same species as that species 2 in my draft genome. So it was nice that this metagenomic environmental sampling was lining up with the culturing. And so, so the results are coming together to support one another. And so that sort of leaves us with where we stand right now with the toxins. We've got a pretty decent handle, at least having confirmed evidence that Microcoleus is producing toxins in the watershed, and we now have a couple cultures and draft genomes of those, but we know there's also more to the story. And so this is primarily information that Rich Fadness, who I just mentioned, shared with me. So he has continued to monitor and is out there each summer, and so he frequently detects nodularins in the watershed. We also collect data on cylindrospermopsin, which is infrequent but sometimes present in the mat, so we don't really know who's producing that. And then once again, saxitoxin is sometimes infrequently detected in the cyanobacterial mats. And here I've included a couple other images of taxa that we know are present in the river, but don't make as large of proliferations as the Anabaena and the Microcoleus. And actually, as I said, the exception would be nostoc. We can get a lot of nostoc in the river, but we haven't really focused on that for cyanotoxins yet. But we know we have cylindrospermum, we know we see nodularia, we know we have cytonema patches, and we also can have oscillatoria. So we definitely still have more work to be done. And so on that, one of our efforts to do more work occurred in summer 2018, and that was when we were fortunate enough to be able to host Susie Wood and some other scientists from the Cawthorn Institute. So this photo on the top here shows Susie Wood in the white vest and the white hat, and then Laura Kelly, who I mentioned earlier, is in the blue, and Jonathan Puttick, who is a cyanotoxin chemist, and Robert Matheson came over here as part of a grant that was received through the Cawthorn Institute. And one of the goals we had was to do some additional sampling to look at the variation in anatoxin concentrations, and also to do more investigation into who might be producing the cyanotoxins. And so we wound up targeting Microcoleus, Anabaena, and then also Cladophora samples and we visited 4 different sites in the Eel River watershed, and then we also got to go to a site in the Russian River watershed, which is the watershed directly south of the Eel. And this has been in the news right now because the big fire burning in California is I think, very just south of that Russian, that number 5 site. So the Russian River watershed is under a little bit of duress right now. So some of the results that came out of that, one of the great things about getting to partner with Susie and Jonathan was they have a great analytical capabilities. And so Anatoxin, there are 4 variants of Anatoxin. There's Anatoxin, homoanatoxin, dihydroanatoxin, and dihydrohomoanatoxin. And so this bar chart here shows the different proportion of those different variants or congeners in our different samples. And we didn't really have great data on this. In the past, we've used ELISA, which just gives you some estimate of bulk anatoxins, or we've just been able to measure Anatoxin A. And so it was very informative to realize that pretty much in all the samples, it was about 50% Anatoxin and 50% dihydroanatoxin. And so it sort of raises questions on if previous measurements and metrics might be underestimating Anatoxin production because we weren't factoring in the dihydro component. Sort of the other data set that supports this was also pretty recent and came from a paper that came out last year, and this was a toxicological study where they actually found a more extreme distribution where it was more like greater than about 95 to 99% of their anatoxins were all dihydroanatoxin A, and that came from extracts from the Russian River. And in the sort of traditionally the literature had suggested that these dihydroanatoxins were degradation products, but in the last few years there's increasing evidence that it's not a degradation product from Anatoxin, but that some strains are actually producing the dihydro variants. And so there's definitely a lot of motivation to do more research into the toxicology of the dihydro variants and understand more about their production and dialing in the analytical methods, or making these analytical methods more widely available, the work that Jonathan Puttick and Susie have been doing. Another great resource that the Kiwis brought was this metric called Anatoxin quota, and this is something that they've published a few times now. I've highlighted a couple of publications there, and so the idea here is a way to kind of get a handle on the different, knowing that there's multiple genotypes within these mats, is taking your toxin data and standardizing it by your qPCR data, your molecular data. And so the way that works is you basically wind up with micrograms per toxin per dry weight mass over gene copies per dry weight mass. And when you do the algebra, you can cancel out the dry weight, which gives you picograms per toxin per cell. And that's because there's only one of the Ana-C genes in each cell. And so for every gene copy, you can use that as a proxy for an actual cyanobacterial cell. And so this is a way to better standardize your sample for the number of toxin-producing cells that are actually in your mat, since we know that can vary over time. And so here is the first part. On the left is the toxins data coming off the LCMS, and then on the right panel are the Ana-C gene copies coming from qPCR. And often we've usually just looked at the ELISA data, and there's about in our results, there's about a 5 to 7-fold variation among the sites when you're just looking at the Anatoxin concentrations. And then here is the quota data. So now we've standardized everything, and it's at picograms of toxin per cell. And what happens is you compress the variability, and so what that means, and change the trend. So you can notice that site number 5 from the Russian River was the second highest, or the highest site when it came to the Anatoxin qPCR value. But in terms of quota, it actually turned out that all those toxin-producing cells weren't producing that many toxins in that site. So it's a useful metric, and it supports, it was encouraging to see that these results support the work that they've done in New Zealand, which suggests that the variation we see out in the environment is primarily explained by varying abundances of toxin-producing genotypes. So when we see really toxic sites, it's usually the fact that there's more toxic genotypes in that sample rather than just a really potent strain that's producing toxins. Now, here's the cryptic part. This was some of the more surprising results we had. So this here just shows the LCMS results for Anatoxin. And what was intriguing here was this is sites came from our site 3, and you can see the colors represent different genera. So this was sampling Anabaena growing on Cladophora. And so the yellow, and then these images show what that looks like. So for the yellow samples, what we did was we targeted one of those conspicuous Anabaena-dominated patches. But you can see that there's a lot of just Cladophora that's growing in there where there is no conspicuous green Anabaena growing all over it. So that's what we mean when we say a Cladophora mat. There was no visible Anabaena on it. But both of those were picking up Anatoxin. So it was surprising to see that the Cladophora, even though we didn't see any cyanobacteria in there, was still had as high a cyanotoxin concentration as the conspicuous Anabaena samples. And the Anabaena was less surprising because that previous work I'd shown you, we already knew that the anatoxins that there were anatoxins within the Anabaena mats. So then what we did to get a better sense of the production was we sequenced the Ana-C gene, and so that here shows the Anatoxin gene cluster, and so Ana-C is there towards the left, and it's actually the gene that starts the whole Anatoxin synthesis process. It all begins with Ana-C. And when we sequenced that, we found that of the 17 sequences, that they were all 100% identical to one another. So that means, there wasn't a lot of variation there, likely coming from the same organism. However, 282 base pairs is not a super long section to be sequencing. But what was very surprising was that when I compared these sequences to those Microcoleus draft genomes that I had constructed, the sequence was 100% identical to the Microcoleus as well. And so down in the bottom, you can see just a little snapshot of an alignment with a couple different genomes. And in red, I've highlighted the sequence from this study, and then also the Microcoleus genomes. And you can see that they're identical for that stretch. And so that held true for the entire 282 base pair length of it. After seeing that, I went and downloaded more reference genomes from both organisms in the Nostochales. So this was like a Phanozomenon, Cuspidothrix, and Anabaena, and then also more Oscillatoreales. And then I lined them and built a little tree, and you can see that the sequences, if it's an Ana-C gene coming from Nostochales, it's quite different from Oscillatoreales. And so it's pretty much about like an 80 to 90% nucleotide difference between these two clades. The yellow star there shows the sample from this study, and then you can see that it's 100%, or the sample right below that, that PH 2015 is one of my Microcoleus genomes, and those two are 100% identical. And so basically what we concluded was that this seems to suggest that the Anabaena might not be the ones producing the Anatoxin, but it might be Cryptic Microcoleus living within these Anabaena assemblages that are producing the toxins. So even though the Anabaena aren't the dominant or visible cyanobacteria, they might be the toxin producer. And we don't right now have great evidence for Anabaena producing Anatoxin. So these other bullet points, we did have 2 isolate cultures from some of that work with Rosalina Stancheva, and neither of those gave any Anatoxin detections. And we also have 2 draft Anabaena genomes, and we found no Anatoxin genes within those genomes from the North Coast. I went back as a result of this and looked back at some of the microscope imagery I've collected over the years. And so here's another example of what the Anabaena can look like. And I did find some images where you can come across different Microcoleus or Oscillatoreales species or organisms within the Anabaena mat. And so I pretty much knew that, but had never, but they're never at high abundance. And so I also went back and looked at a couple cell counts I had done. And so back in 2014, we'd done some cell counts on some Anabaena mat samples. And in the top there, so we had 31 samples, and I basically it looks like we observed Microcoleus in 24 or 77% of those samples, and it was absent in 23%. So it does seem like Microcoleus is quite common, a member of the Anabaena assemblage. Here's a relative abundance, and also just to note that these are all actual cell counts. These are not biovolume. So you'll see that there's a lot of diatoms in there, and that's just the cell counts and not biovolume. But the Microcoleus is present. But then this histogram shows, if you look at those cell abundances, a lot of the times it's below 5% of the cells in the sample, and for many of the samples, it's well below 1% of the cells in the sample. So it doesn't seem to be a large part of the assemblage, but it is present.
Quick question?
Yeah, sure.
Yeah, that slide. Wondering about epithemia. So, of course, epithemia associates with the Cladophora, and you've probably seen the endosymbionts, the cyanobacteria growing inside epithemia. Wondered, because we have a lot of that in Oregon too, do you think that those coccoid cyanobacteria produce some toxin?
I don't think it's the epithemia endosymbiont. I could go back and double check. That genome was also sequenced a few years ago, and I don't believe they mentioned any anatoxin genes. And also in my results, when I was searching for the genes, I had parts of that genome, and none of the gene hits ever were associated with that, so I doubt it. You'll also see that I have, like, in this slide, a cyano other category. That's a big lumping of a lot of Leptolingbia, some other coccoids such as Aphanosiki. So definitely we have other, some of those small, like weedy or single-celled ones in these mats. Once again, I think the fact that Ana-C gene came, that sequence identity matched up so well with the Microcoleus one. I think that's pretty strong evidence that it's likely the Microcoleus. But it's still not, that's not a bulletproof answer. I mean, ideally, it'd be great to get more like pure cultures in there. But and then you also just highlight, how many species there are and how much more there is to understand in terms of toxin production. Does that answer your question, Kurt?
Yeah, it does. Great, thank you.
Yeah, sure. Thanks. Okay, so that's kind of the conclusion on the Microcoleus and Anabaena bit. We're going to switch gears a little bit to nodularin, and so once again, we found. So what we did was we were measuring microcystins and nodularins, which are both liver toxins, and we wound up lumping these together, partly because we had very low microcystin levels, and also because the qPCR, when we're calculating the quota, the PCR amplifies both genes because these genes are similar, so you can't really distinguish it when you're trying to calculate those quotas. But here we once again see that it wasn't the Microcoleus mats, but it was the Anabaena and Cladophora mats where we detected the highest nodularin concentrations. And we were, once again, able to get the sequence of the NDAF gene that was detected in these mats, and that gene had a 99.7 nucleotide identity to Nodularia spumigena. And so far, globally, there's been very few species that have been linked to nodularin production. And what was surprising about this, too, was that over the years, I've definitely come across nodularia in these mats. So here's some images that I took back in 2013 from 3 different samples, and you can see that there's nodularia in there. The Nodularia spumigena is often associated as a brackish water species, and this is way up in the headwaters of the Eel River and in a very good water quality condition. And so this might also answer why Rich is getting such frequent nodularin detections on his spat, is we might just have all this cryptic nodularia growing in these Cladophora assemblages that's constantly just producing nodularins, even though you never actually see big conspicuous mats of nodularia. In all my sampling, I've never come across a mat that was dominated by nodularia. And then also it's surprising to see all this up in the freshwater, and I think it'd be great to do more molecular work to get a better sense of the species identity of these organisms, because we haven't done that yet, and then we could maybe see if we do actually think they're nodularis, Bouma-Gregson, or if they're a different taxa. So where we're going in the future here, this summer, to try and follow up the work on the Anabaena, the cryptic producers in Anabaena, I've been working with Rich again, and we collected 25 mat samples from about 7 different rivers. And so what I want to do is try to expand the data set that we collected with the New Zealanders and try to get a better handle on if Microcoleus is always the producer in these Anabaena mats, or if we do actually have an Anabaena producer. So we've already done ELISA Anatoxin work on these samples and a cursory microscopy to look at dominant and subdominant species, and that's what this bar chart is. And so you can see that of these samples, I've listed the dominant and subdominant species in them, and the majority of them were Anatoxin was detected with ELISA. The next step is we were going to want to do 16S high throughput Illumina sequencing to get a whole sense of the, use molecular tools to get a sense of the diversity in these assemblages, and then also do the PCR and sequencing of the Ana C gene to match up the gene sequences with the databases and see if we do ever get a gene sequence that is more closely associated with a Anatoxin rather than a Microcoleus. So that's the work we're going to be doing this winter, is this molecular work. So in conclusion, we basically continue to support the idea that multiple cyanotoxins are produced within wadable streams, and that wadable streams are our source of toxins. We've shown continued this cryptic diversity and the strain heterogeneity within the cyanobacterial species, with some species and strains producing toxins, other not. It's great to have the work in California support the work in New Zealand, which is showing that the toxin quota really helps standardize the toxin production, and that it provides a lot of evidence that it's the genotypes that's really driving toxin concentrations in the environment. The dominant biomass may not be the dominant cyanotoxin producer, and a lot of questions about nodularian diversity and nodularian production based off of this work. And in terms of management, one of the things that this has had me mulling over is, in terms of a dog or the public, they don't really care who's producing toxins in the mat. They just care if they eat that mat, are there toxins in it. So, how this work, it doesn't necessarily need to change the way we talk about the production of things. However, the messaging that was most surprising for me was seeing the toxins in the Cladophora, because now I feel like the question is, well, do we need to just tell people avoid all algae, even if it's a filamentous green algae that we know can't produce toxins? Might there be cryptic cyanobacteria in that mat, or should we just continue, or should we be focusing people in on the known mats of toxin producers, such as, well, just don't eat Microcoleus? And then also we need to continue a lot more work on anatoxin toxicity and those dihydro variants and the toxicity there. I think that's going to be an important next step for thinking about human health issues. So, acknowledgements. The Cawthorne Institute was awesome, working with Susie and JP and Kelly. The paper has been submitted. We just already submitted. It's already been through a round of review, and so that paper, where Laura Kelly will be first author, will hopefully be out in the next few months. So keep an eye out on that, and I can share that with the group. And a huge thanks to Laura for taking the lead on writing that up and synthesizing those results. We had support from the regional water boards, Rosalina at Cal State San Marcos. A lot of the PhD work I did was supported by the Reserve System, NSF, and the EPA. So huge thanks to all these groups for funding this work. And I can take any questions, and I apologize if I went a little bit long too. So thank you, everybody.
Does anybody have any questions for Keith?
Yeah, I have a comment.
Sure.
This is Kurt Carpenter again from Oregon. So really awesome presentation. So glad to be on the call here today, and thanks to Holly Bellringer for letting me know about this group. I'm doing really similar kinds of research in Oregon on a lot of these cascade drinking water rivers, and you mentioned that you've got nostoc, and we have a lot of nostoc. We've got both the Parmeloides and the Spongiforme especially, and the benthic samples that I've been analyzing with ELISA, oftentimes I'm finding all 4 major cyanotoxins, so Anatoxin, saxitoxin, microcystins, and cylindro as well. So I encourage you to look at the nostoc, but I really appreciate your work, Keith, and that was mostly just a comment to look at the nostoc. One of the things I'm finding with the nostoc in particular is so I'm doing the colonies and then also spats. Most folks are aware of the spats. That's a great thing. And then also doing plankton net toads in the rivers. And then under the microscope, I see a lot of nostoc balls, really tiny nostoc balls in transport. And then those samples are turning up, all 4 of these toxins as well, and including saxitoxin at a high frequency of detection at some locations. So just some ideas to consider. Thanks again.
Yeah, thanks, Kurt. That's awesome to hear. It sounds like you've got a great data set. And I know talking with Susie that they get a lot of microcystin production from nostoc in the New Zealand rivers as well. And I didn't focus on the nostoc initially, partly because of its kind of more like from the public health angle, where it tends to be more tightly attached and up a little bit more in the headwaters, and less associated with recreational sites. But I sort of regret that decision now, it's clear that as the data comes in that they can be a lot of producers. So I know Rich has collected a lot more nostoc samples, so he also has data that we should it'd be great to compare some of the data he has with your data as well.
Sounds good. I'll be in touch.
Yeah, that'd be great.
This is David Fair. I'm a toxicologist with Oregon Health Authority, and you might have already covered this, but I was wondering how you were detecting anatoxin, and especially the differentiating between the hydro variants of anatoxin. And for example, like most important for public health would be whether the activity-based ELISA type plates that are activity-based would capture both the anatoxin and the hydro variants.
Yeah, so this was all with tandem mass spec and differentiating those. Some of those, my understanding is that the ELISA does capture more of the questions. I don't know if Jonathan Puttick is on the call right now, but he is the analytical chemist to talk to when it comes to Anatoxin. I don't know if Jonathan, if you're on and you want to chime in at all.
Yeah, I'm here. So like you said, Keith, we're completely right. We're using LCMS to be able to differentiate the different anatoxin congeners. And with the ELISAs, you don't sort of get that distinction, and I'm not sure if anyone's done any work on cross-reactivity with the anatoxin ELISAs between anatoxin A and those dihydro variants. So what I mean there is, if you have dihydro anatoxin in your sample, would it actually be getting detected by those ELISAs, or are they so specific for anatoxin A that it doesn't detect it?
Thanks, JP. I know that talking with Rich, who's submitted some samples for just Anatoxin A LCMS as well as the ELISA, he has come back with non-detects from the LCMS, but detects from the ELISA, and he thought that might be a result of the ELISA picking up the variants, especially knowing that we do see a lot of this dihydro now that we have evidence for that. So, JP, I don't know if you think that is good evidence for that or not, but that's at least one thing I've learned from Rich and the data that he's gotten when he's submitted to both just a single LCMS, so not tandem LCMS, single LCMS and ELISA.
Yeah, that does suggest that you might be picking up the dihydro anatoxins, but it would be interesting for someone to do if they're using anatoxin ELISAs, and we're able to get some of dihydro congener. yeah, I guess it's something that we just need to keep chipping away at and be able to understand that.
Yep.
Okay, are there any other questions on the phone for Keith? So Keith, I'm not sure if you're able to look at the chat box or not, but there were a few questions. What I might do is see if we can grab those questions before we close out the WebEx meeting and submit them to you, and perhaps you could, write up a little response that we could share with the work group.
Sure, I do. So I'm looking at now, but the intra versus extracellular, just a little bit. The mat samples would be a bulk intra and extracellular, but the spat samples and I've collected just a couple filtered water samples, that would be extracellular. So just a little bit of data on that. Primarily, we think of them as being intracellular, though. That was Cale Clausen from Oregon DEQ. But yeah, that sounds great. I can go through other comments too.
Okay, great. That would be perfect.
Christine. Thank you, everybody.
Well, thanks, Keith. We really appreciate you volunteering your time and presenting on the research and the work that you're doing. Hearing the previous item with the survey results and the open discussion between the work group members of where there's a lot of interest for development of subgroups, or especially topics and focus that everyone would like to get more information on. It's really appreciative to hear the research and work that's going on, not only here in California, but elsewhere across the country, as well as other countries such as New Zealand, Australia. There's a lot of work going on in France and Spain, from what I understand, and even Canada. So it's just really great to have this opportunity for the discussion and presentation of research and information. So we're going to move on real quickly to the next item, because we want to wrap this up, get everybody out. And so it's the open discussion, publications, and upcoming meetings. Jade, could you go to the next slide? So really quickly, we want to do a little shout out to our group, and also to two of our facilitators, as well as Rich Fadness. So Margaret Spoo-Chupka and Jade Young, which are two of the co-facilitators here for the Benthic HABs Workgroup, along with Rich Fadness. You heard his name mentioned today. He works in our North Coast Regional Water Quality Control Board here in California. They were approached by Angela, who's one of the, the editors for Lakeline Magazine, and they did a article called A Deeper Look at HABs. I encourage everyone to check it out. It was a really interesting article, and actually it was designed and focused to make people understand that there's more than just our water planktonic HABs that are going on. We do have these benthic HABs occurring in both our wadable streams and rivers, and also our lakes and reservoirs. So the link is available on the slide there, and it's open access on that particular issue, and you can read all about, there's some remote sensing articles, I believe, also in that issue as well. So that was a great job by our co-facilitators and Rich. Okay, you can go to the next slide. So, one of the things that I always struggle with, I'm not an expert, not nearly as much as Keith and some of the other people that I'm listening to on the call. So for me, it's always a learning opportunity, and part of that comes with finding publications, articles that are being published by researchers. And so I know I always appreciate it when someone shares something with me, especially if it's an open access article, because I know a lot of us are limited by potentially getting access to articles and journals. So one of the things that we like to do is just do some shout outs for some recent publications that maybe we've come across that we think you might be interested in. The first one is by Zachariah Smith. It's the Spatial and Temporal Variation in Paralytic Shellfish Toxin Production by Benthic Microcyra Lingbia Walleye in a freshwater New York lake. That is a really interesting article, and so it's looking at a lake in upper New York State called Butterfield Lake. They found paralytic shellfish poisoning toxins and trace levels of Anatoxin A, and they were looking at the environmental and temporal drivers. And so based on the abstract, they said they found distinct differences in the total paralytic shellfish toxin content, filament nutrient, filament chlorophyll, and relationship to the environmental drivers between sites. And in that study, they also developed a multivariate model that explained 85% of the variation in the paralytic shellfish toxins observed that were growing over the season. So that's an open access article found in Toxins. I invite you guys to take a look at that. The second article is spatial and temporal variability in the development of potential toxicity of formidium biofilms in the Tarn River in France. Again, there's a lot of work coming out of France and Spain. So I invite you to take a look at that. Basically, they were looking at formidium biofilms and the production of anatoxin over 2 years. And they found that the biofilms exhibited strong spatial and temporal variations and was correlated positively with water temperature and depth. And then finally, there's another article that Keith Bouma-Gregson and others came out with in 2019, Impacts of Microbial Assemblage in Environmental Conditions on the Distribution of Anatoxin A-producing cyanobacteria within a river network. So again, open access. And if you have any others that you'd like to share with the work group, feel free to send them to us, and we can make sure that we send that information out to our work group members. I know that I appreciate it when I read it in the freshwater EPA Cyano HABs newsletter that Lesley Dungalata puts together. So again, this is an opportunity to share publications. Again, we're out of time, but if other people have stuff to share, just go ahead and send it to me. We can send it out. And if you go to the next slide, there's some upcoming HAB meetings that people might want to be aware of. There's the 10th U.S. HAB Symposium occurring next week, November 3rd through the 8th in Orange Beach, Alabama. There's also a NALMS meeting called Watershed Moments occurring the week after that, November the 11th through the 15th in Vermont. And there's a Water Quality Technology Conference that's also happening next week in Dallas, Texas. Again, if you have any information on upcoming HAB meetings that the work group members would be interested in, please make sure to share it with us so we can share it with everyone else. Go ahead to the next slide. And go ahead, and this is just a wrap up. We're going to talk about our schedule really quickly. So as you heard through the survey, people really like to have that quarterly time frame for our meetings. So our proposed 2020 schedule is sticking on that timeline of January, May, July, and October. Again, we're always looking for speakers, and it sounds like from today's discussion, it sounds like we don't always need to have a speaker. And so we do appreciate hearing that, and we're going to take a look at some other potential models that we can use for meetings. But right now we do have for our January 2020, we have a tentative speaker, Zach Smith. He's one of the authors of one of the papers that I showed you earlier on the other slide. And he would like to present some information on benthic HABs and paralytic shellfish toxins in their freshwater New York lake. So they would be able to provide some information on that from another state here in the United States, which is New York. So again, if you have some research that you'd like to present on, or even if you'd like to talk about your monitoring program or the sampling, I know that one of the questions that one of the work group members had was talking about sampling collection methods. So perhaps we can have a topic discussion associated with that. So I'm sorry that we're out of time. I want to kind of keep on our schedule. So if you do have any other comments, just feel free to send them to us at our email. So again, we've posted material on our Benthic HAB work page. If you have questions to Keith, you have his contact info. And again, if you have anything that you'd like to share with us or be added to our list, go ahead and contact us. Thank you, everyone, and I hope you have a nice Halloween tomorrow and have a nice end to October. Margaret and Jade, do you want to say anything?
Thank you.
No, thanks.
Okay, great. Thank you, everybody.
Thank you.